Hi, everyone. Thanks for joining the Boston Epigenetics Society. This is our June webinar, and we have a great speaker lined up today. I'm very excited for today's presentation. Before we get started, I just wanted to take just a brief minute just to introduce the Epigenetics Society. If you've joined us before, great. I'm very happy to have you back again. If you haven't joined us before, well, welcome. I'm glad you could join us for today's webinar. The mission of the Epigenetics Society is to promote epigenetics and epitranscriptomic research, and really it's to foster connections between academic and industrial scientists. I'm supported by a great leadership team. They're listed over here to the left. In a minute, I'll be introducing Ollie, who's going to be today's MC. And we're also supported by Dovetail Genomics, Active Motif, and Alida Bio. So I'm very thankful for our sponsors. Without them, it would be impossible for us to do this. Also, I want to encourage you to reach out. If you're interested in getting more information or becoming more active, we do have an email address at bostonesociety@gmail.com. And we also have a website. So you can check out our webpage at bostonepigeneticsociety.com. And at the bottom of this page, you can sign up for upcoming information and all of our presentations from the past, and as well as today's presentation will be posted on this website, so you can see all of our former talks. Also encourage you to sign up for our group on our LinkedIn page. So if you go to LinkedIn and then you search for Boston Epigenetics Society, you can find us there. As far as today's seminar, there is a Q&A window, and I encourage you to ask questions during the presentation. We won't pause during the webinar for questions, but we will ask those questions at the end of the webinar. So at this point, I'm going to turn it over to our MC, Ollie Jin from Dovetail Genomics. Thank you very much for joining us.
Thank you, Brian. Hello, everyone, and thank you all for joining us again. We're very excited to welcome today's speaker, Dr. Mathieu Lupien. Dr. Lupien is a senior scientist at the Princess Margaret Cancer Center at UHN and a professor at the University of Toronto. His research has helped reshape how we understand cancer biology by demonstrating that tumor progression is driven not only by genetic mutations, but also by heritable epigenomic variation encoded in chromatin. Over the years, his work has provided major insights into how non-coding regulatory elements are hijacked in cancer, how chromatin states influence cellular identity and plasticity, and how environmental pressures such as hypoxia and metabolic stress can drive tumor evolution through epigenetic remodeling. His research has contributed significantly to the broader understanding of tumor heterogeneity, cancer evolution, and chromatin-targeted therapeutic strategies. Today, Dr. Lupien will present a talk titled From Epigenomic Heterogeneity to Tumor Evolutionary Trajectories. In this presentation, he'll discuss how epigenomic diversity and cell state plasticity shape tumor evolution and dissemination, along with emerging strategies to track these processes through integrated analysis of chromatin state, transcriptional programs, and lineage information. We're thrilled to have him with us today and think this will be a fascinating discussion for anyone interested in cancer biology, epigenomics, and tumor evolution. Without further ado, please join me in welcome Dr. Lupien.
Excellent. Thank you very much for the invitation. It's a real pleasure to be here today and to have this opportunity to present this webinar. Those of you that have had the chance to see work from my lab in the past will probably have a little smile on your face when you read the bottom left sentence here, which is DNA holds the words and chromatin tells the story. That's truly the premise through which most of the research that's ongoing in my lab is being led, and that we recognize the importance of DNA sequences, of mutations, but at the same time, the need to be able to position those mutations, those DNA sequences in the right chromatin context. And so with that in mind, it will come to no surprise that, again, we're heavily focused on understanding the basis of cancer through a chromatin landscape. And so just to put the perspective and the terminologies in place for the rest of the presentation, as a reminder, the DNA in our cells is packaged into chromosomes. And if you zoom into these chromosomes, you can actually identify specific structures, predominantly the nucleosome, which are composed of a series of 8 proteins around which 147 base pairs of DNA is wrapped to define what we call nucleosomes. And the nucleosomes are pretty much the basic unit of the chromatin. And so you can study and measure differences in chromatin states and chromatin identity pretty much at this single nucleosomal resolution. Now, there's a couple of concepts that I want to put forward that I think are critical to remind all of us as we think of diseases such as cancer, but of course, other diseases, when we're trying to focus specifically on features that are inheritable. As a reminder, when a cell divides, the material that gets replicated is not just the DNA sequence. This figure here is meant to represent the parental strand on the far right and the leading and lagging strand on the far left. And the key take-home message from this is that, yes, this replication fork occurs, in which case you have effective DNA replication taking place. But at the same time, the information, the chromatin-based information that's found in the parental strand is still shared and maintained in this replication fork. So the DNA methylation will, of course, be still associated with the DNA sequence itself, and machinery will come by to ensure that in the de novo strands, the proper DNA methylation patterns are being added. And from a nucleosome standpoint, from the proteins that are found within each nucleosome, half of them go to one strand, half of them go to the other strand in order to be able to maintain the information that they harbored in the parental strand to ensure that this replication process is as efficient and with high fidelity as possible. And so not only are we replicating the DNA sequence, ensuring that your daughter cells end up with the same DNA sequence, but as well, you're replicating the chromatin, ensuring that any given piece of DNA is still found in the same chromatin context that it was in the parental sequence. The second aspect that I also value in reminding is this notion that when the cells are about done with the division, there's this segregation step that takes place. And while we've in the past put extraordinarily emphasis on the notion that DNA segregates in a Mendelian fashion, I'm just reminding you here with these images that the material that's actually segregating in this anaphase time point is not just the DNA sequence, it is the chromatin itself. And so both the DNA sequence differences that would have been picked up as errors in the replication process, as much as errors that would be introduced in the chromatin state composition, would be segregating in a Mendelian fashion. And that's the fundamental basis of the principles that are required to ensure fidelity in cell identity over cell division. And so if one really cares about understanding how plasticity operates in a disease context, well, one should not just rely on looking at DNA sequences, but definitely heavily rely as well at looking at chromatin composition. And that's exactly the premise according to which we study chromatin, not only DNA mutations. Now, the chromatin, in contrast to the DNA sequence, is much more complex than a series of 4 options, right? For the DNA, you have A, C, T, and Gs. Now, for the chromatin itself, it can come in one of tens, if not hundreds of different chromatin states. And so what this image is meant to depict here is from the collection of ChIP-seq data sets across a series of histone acetylation and histone methylation, only a subset of the types of post-translational modifications one can see on the histones, they come into different patterns. And so each line corresponds to a different pattern of histone modifications that are found together across a sufficient number of DNA sequences across the genome. And interestingly enough, some of these patterns are typically seen around regions of the genomes that act as promoters. Other patterns are typically seen at regions of the genomes that act as transcribed areas of the genome, so genes, non-coding RNAs, microRNAs, and the likes. Other patterns are typical of distal cis-regulatory elements, primarily enhancers, and so on and so forth. And so being aware of these patterns allows you to annotate the human genome very effectively so as to be able to distinguish whether a given piece of a sequence is currently utilized in a given cell as a promoter, as an enhancer, being transcribed, or plain and simply being repressed. And so one has to keep in mind those types of alterations in order to really be able to start assessing when disease occurs, what changes are happening in the chromatin state itself. And so what my lab heavily focuses on is specifically in searching for these regions of the genomes that change in chromatin state between, for instance, normal tissue versus cancer, or primary tumors versus recurrence or metastatic tumors. And whenever we find such regions of the genomes that change in chromatin state, not necessarily with a change in DNA sequence, we refer to these regions as chromatin variants. Now, chromatin variants, you might not be extremely familiar with that term, but you're surely familiar with the terms such as epi-lesions, epi-mutations, DAR for differentially accessible regions, DMRs for differentially methylated regions. Well, all of these subdivisions, more or less, can fall under the premise of chromatin variants to the same extent as different types of mutations and or inherited variants can fall under the premise of genetic variants. So we use the terminology chromatin variants when referring to any area of the genome that changes its chromatin state composition in normal versus disease and primary versus recurrent status. Just to give you a few case examples of what we mean in our hunts for chromatin variants. So this is looking at data from normal tissue biology that was published a few years back by Wouter Mühlmann, as well as John Stamatoyannopoulos, where they specifically looked at a chromatin state defined as accessible chromatin, so regions of the genomes depleted of nucleosomes, where DNA is in principle free to interact with DNA binding proteins, such as transcription factors, RNA polymerase II, as much as DNA repair machinery. And what was done here was to look at chromatin accessibility across a large range of different cell and tissue types, in this case, a subset of them being labeled on the right side of this image. And each track is depicting the signal of chromatin accessibility. Whenever a peak is detected, it implies that this is a region of the genome that is depleted of nucleosomes, so an accessible chromatin in the genome. And we're looking at a very small area of the genome here, as you can see from the metric specified at the top of the figure. Now, the key take-home message that I want you to take in from this figure is that you can identify areas of the genome, such as this one on the right side, on the left side, which is preferentially accessible only in a subset of the cells and tissue types that were assessed. And if you go all the way down to the right side, you see that this peak of accessibility is restricted to B cells. In other words, this is a chromatin variant that's pretty much specific to B cell, i.e. it harbors a DNA sequence that's most likely essential for B cell identity. Of course, you have other regions of the genome, such as this one that's depicted in the middle, that are accessible across all cells and tissue types. And so these would be typical of essential genes, associated chromatin accessible sites, i.e. regions of the genomes that do not have cell type specificity, but rather are critical for cells to exist and survive. Okay, good. So that's the premise of looking for chromatin variants. We can find those that are specific to our tissue of interest or cell types of interest. And the underlying principle is that the DNA sequence that's found within these chromatin variants has greater value for that cell type than it does for any other cell types. Okay, good. Now that's in the context of normal biology. What about cancer? Well, we actually have to go back to 1980s to be able to actually identify the first set of manuscripts that reported chromatin variants in a cancer setting when compared to normal tissue. And this was specifically done in those days looking at DNA methylation. First, Melanie Ehrlich, which actually in the Science publication in '82 put the concept forward with listing data that was supportive of chromatin variants based on DNA methylation existing in cancer. And then in 1983, coming up with this publication with actual data showing and identifying chromatin variants. In parallel, in the same year, Andrew Feinberg and Bert Vogelstein came up with evidence of chromatin variants in cancer based on DNA methylation. The data that I'm portraying here on the right side actually comes from the Feinberg publication, looking at normal tissue, cancer, and metastatic tissue, isolated from colorectal origins or lung origin, using restrictions enzymes that would have DNA methylation sensitivity or not. The premise here is looking at the percentage of hypomethylated fragments. You could see that in normal versus cancer, it actually went up in the cancer setting. Down at the bottom, normal versus cancer, and then cancer versus metastasis. Again, significant differences in the degree of DNA methylation being depicted at these regions based on enzymatic-based activity. So that was in 1983, a long time ago, right? Well, it actually took a few more years before being able to report the similar biology from histone modifications. So this came in 25 years later in 2008, work that was conducted by myself while a postdoc in Miles Brown lab, looking at breast cancer versus prostate cancer tissues or cell types, and assessing the reason as to why a specific factor, in this case FOXA1, that has the same DNA binding domain, could actually occupy different regions of the genomes in prostate versus breast cancer. It turned out that the reason was for the presence of differential histone modifications, H3K4 mono and dimethylation, being required at the site, enabling FOXA1 recruitment. Now, since 2008, numerous studies have further expanded on this concept that histones or nucleosomes are going to be a mean to identify chromatin variants. A beautiful study in Science from Ryan Corses a few years back, comparing chromatin accessibility across over 400 tumors collected through the Cancer Genome Atlas initiative, identified a series of chromatin variants specific to given cancer types. So here at the top, we're looking at colorectal cancer. At the bottom, we're looking at kidney cancers. There's a nice area here that only has signal of accessibility in colorectal cancer, lacking accessibility in the kidney cancer cases. So a chromatin variant specific to colon, and vice versa on the far right side, an area that has no accessibility in colon, but clear accessibility in the kidney tumors, i.e. a chromatin variant specific to kidney cancer. Now moving forward, in 2014, in collaboration with Mike Taylor, actually a work led by Mike Taylor on brain tumors, specifically on pediatric ependymoma, identified the first case example of a cancer arising free of driver mutations and instead relying on chromatin variants, relying on these changes in the chromatin state picked up in cancer versus normal. So quickly speaking, here on the left side, we're looking at results from whole genome sequencing or whole exome sequencing in pediatric ependymoma at the bottom versus a collection of other cancer types. And you can see that both the red line, which is the mutations identified per megabases, or the blue histograms, which is the number of significantly and recurrently mutated genes, it's pretty much down to zero in ependymoma. No evidence for driver genes, driver mutations in this cancer type. And it's a pediatric, not enough time to accumulate mutations, in contrast to other solid tumors. However, in this case, looking at DNA methylation, conducted across a collection of samples, led us to identify two subgroups, the group A and group B ependymoma, one of which showcased increased levels of DNA methylations across hundreds of CPG sites. In other words, chromatin variants picked up from a DNA methylation perspective. And interestingly enough, in the manuscript, we had functional studies using model systems, but subsequently a clinical trial was initiated. And this is a case example of what was observed in some patients. Pre-treatment, the blue area depicts the tumor. 3 months after DNA methyltransferase inhibitor treatment, you can see that the area in blue has significantly decreased. This is looking at the individual tumor from one angle, looking at the same tumor from a different angle, again, depicting a significant reduction in the tumor mass when treated with epigenetic therapy, in this case, using DNA methyltransferase inhibitors. And shortly afterwards, familiar name, Andrew Feinberg, also identified pancreatic cancer progressing to metastasis as being driven by epigenetic changes independently of mutations. And of course, since then, many other studies have moved in that direction. Beautiful work from Brad Burstein a few years back in gliomas, identifying a combination of both genetics and mutation, genetics and epigenetic changes in driving the formation of gliomas. And for those of you that have seen this work, you'll love it. In 2024, the lab of Giacomo Cavalli demonstrating using a Drosophila system the ability to induce cancer fate, plain and simply by playing around with the activity level, as opposed to mutations, activity level of enzymes involved in the polycomb complex. Okay, good. So again, a little bit of history, right? Putting things in perspective with all of this work between 1982, 83, when the concept was first put forward, it took over 40 years for epigenetic reprogramming, so for these concept that chromatin variants can be contributing to cancer, to be formally recognized as an element, an enabling characteristics within the hallmarks of cancer, as reported by Hanahan in 2022. Okay, good. Now this work is critical in order also to better understand the mechanisms of tumor evolution. It's one thing to be part of the hallmarks of cancer, but that's not necessarily the end point. One needs to be able to take advantage of the knowledge acquired to better understand the principles of tumor evolution. And where epigenetic changes, where chromatin variants can truly have a significant impact and where we need to pay attention to them, is being a new source of heterogeneity. Like it's clearly established that genomic instability and therefore genomic heterogeneity is a driver of the most aggressive forms of tumors. Well, to no surprise, if in addition you can increase your heterogeneity and population identity from an epigenetic standpoint, then you further increase your likelihood of being equipped to thrive in changing environments. So epigenomic heterogeneity being a feature of interest for us to study. A second aspect is, of course, that by manipulating the chromatin state, one can effectively reprogram cellular identity and therefore allow cells to transition from one fate to another in a system that's very much more malleable, if one can say so, than for mutations. And then another aspect that one needs to keep in mind is that from an epigenetic standpoint, it is also possible to track prior stresses, prior insults, which will be embedded within what we call epigenomic memory. So signatures from the past that are still embedded within the chromatin landscape of a given cell, a given normal cell, or a given cancer cell. Perfect. So what I'd like to do now is jump into some new results and specifically focus on prostate cancer as a case example in which we can study chromatin variants to better understand the mechanisms that enable tumor evolution across both development and progression. Now, prostate cancer, as for many other cancer types, right, has a series of different steps to engage in a pre-malignant state, to then engage into a full malignant state before it metastasizes. And what I'll discuss first is this initial phase of malignancy, when cells have transformed and are on the path to exiting the primary site, invading the lymph nodes before embarking in a full-fledged metastasis process. Looking at chromatin variants, looking at the epigenetics, allows us to more or less assess tumor heterogeneity. Now, when one talks about tumor heterogeneity, one often refers to intra-tumor heterogeneity, but the reality is that there are many different layers to heterogeneity that we have to keep in mind. Interpatient heterogeneity is one, intra-tumor heterogeneity is another, multifocal, or if you prefer interfocal, interfoci heterogeneity is a third, and then intertissue, primary versus metastatic sites, is a fourth layer of heterogeneity that one can study. And so we decided to embark on a project to study the relationship between epigenetic heterogeneity across all of these four different layers of heterogeneity. This work was led by Tina Keshavarzian and Kia Furlano, and specifically focused on assessing the heterogeneity within a collection of 5 primary prostate tumor samples. We selected 5 samples from different patients and went in great depth at characterizing these 5 patients by being able to sequence at single cell resolution the nature of these cells from a chromatin standpoint, with the ATAC assay being embedded within the single cell multi-ohm assay, as well as gene expression, RNA levels. From each of these patients, we had multi-foci collected such that we could not only assess the intra-tumor heterogeneity, but interfoci heterogeneity at the same time. So this first figure here, the UMAP in the middle, actually is color-coded based on patient of origin. And as has probably been made obvious to you, there's a high degree of patient specificity in the localization of individual cells within this UMAP. And so to no surprise, there is a high degree of inter-patient heterogeneity, as anybody else has depicted in the past when working with single cell data. Here, what we've done is actually annotate these cells such that one can distinguish malignant epithelial cells, so the cancer cells in green, versus all the normal cells that could be found in these same tumors. So B cells, macrophages, T cells, and so on and so forth. And what you may be in a position to observe is that those colors of cells that are normal in identity cluster predominantly in the center of this U map, an area where you actually have cells coming from all patients. And so this is depicted here on the right side image where we quantify the degree of patient intermixing. The higher, then it means that there are more patients contributing to the collection of cells composing one population of interest. And so as you can see for the normal populations, we have typically normal populations that are composed of cells coming from all patients. In contrast, the cancer populations are extraordinarily patient-specific, extremely low to no patient intermixing. So this is an important feature to keep in mind. Of interest as well, although the sample size is small, the other group of normal populations down here at the bottom that show no patient intermixing, that are patient-specific, are B cells. And we can talk about this subsequently if you want in detail. Okay, so high degree of interpatient heterogeneity, specifically focused on the cancer cells. Second question, what about the multifocal heterogeneity? What about the intra-tumor heterogeneity? So here in this figure, I'm zooming into our 5 patients, showing you approximately where the multifoci biopsies came from that were processed by single-cell multiome. And so in one of these patients, we had 3 biopsies and all the way up to 8 biopsy in this patient to the yellow patient. All of these were processed separately so as to be able to identify intra-tumor heterogeneity as well as interfoci heterogeneity. So from these data, here I'm depicting you the results from one of these patients. What we first decided to do was to cull populations within this collection of foci that were processed by single-cell multiome to see if we first identified more than one cancer cell population, and if so, was it most variable within site or between sites, within foci or between foci? And so here I'm depicting you all the populations that were identified. Again, in green are the cancer populations, and the other colors are in the normal population. I'm showing you from left to right, attack at distal sites, attack at promoters versus expression. So chromatin accessibility at distal sites, chromatin accessibility at promoter versus gene expression. In the degree to which either of these assays, chromatin accessibility or gene expression, could discriminate these populations. In agreement with prior publications in other cancer types and other studies, what was found was that the attack signal at the distal sites was providing the highest degree of resolution at discriminating these different populations. Not the gene expression, not the promoter signal, but rather the signal found at the distal sites. The second observation that comes from this analysis is depicted in the middle panel here, which is focused on the signal or the proportion of cells from any given population versus the different foci that were studied. Okay, so again, the foci here are different biopsies. And if it's bright red, it means that pretty much all the cells from a given population come from a single foci. The paler the red, the more heterogeneous across foci the population will be found. So for instance, I'll focus here on the number of normal populations. What you could see is if you go into the foci distribution, the red is relatively pale and found across all foci. So it tells you that whatever that normal population is, it's found in just about every foci. It doesn't really change in identity from one foci to another. However, now when we look at the green populations, which again are the cancer population, the malignant epithelial population, what you'll see is that they're typically associated with a single foci. So in other words, there's always a very red intensity red assignment for each of these populations to one of the foci. It's very rare to find a cancer population that is actually distributed across all the foci. And so what this is reflective of is that the highest degree of heterogeneity between the cancer populations lies in whether they're coming from foci 1 versus foci 2. So foci-specific heterogeneity is the highest. The previous slide was for one patient. Here I'm showing you the results from the subsequent 4 patients. Again, the same principle. The only instances where we see malignant cells being found across all foci is typically when that malignant population has a high score for cell cycling. This is the results that are summarizing all of the data where we're measuring foci intermixing. So again, most of the normal populations on the right side are found across all foci, all biopsies from a given patient, while the cancer population tend to be foci specific. Excellent. All right. Now, we talk about foci-specific populations, but we need to put that into relative context. So here what we're doing is showcasing the degree of similarity based on a Pearson correlation that exists between normal population, one versus another, and then cancer populations, one versus another. And so as you can see here, this value of Pearson correlation that lies just above 0.4 is typical of cells that are very different in identity. So B cells versus T cells, T cells versus macrophages, and so on and so forth. And then in contrast, the cancer cells, despite being foci specific, show between a 0.6 and a 0.8 or above Pearson correlation in identity. So there's definitely a high degree of similarity across those cancer populations. One that is definitely less than what one would observe when comparing cells of completely different identity, again, such as B cells and macrophage. Now, could this foci-specific population of malignant cells be justified based on their genetics? In other words, is it that each foci has a unique genetic composition, and that's the genetic composition of that foci that's driving the spectrum of population of cancer cells that can be found in one foci versus another? To address this, we modified actually a tool to identify or to report CNVs from single-cell multi-omic data called NumBat, so as to be able to call CNVs not just from RNA, the premise for which NumBat was first developed, but also from the ATAC data, combining the two data sets. And so here we're looking at one patient where we're assessing the presence, absence of clones, genotypes, and we could identify 8 different clones. And you can see the degree of similarity or differences that exist between those based on regions that are amplified or deleted. And then here in the middle, we're depicting the distribution of these clones across all of our foci. And for the majority of them, they're found in every single foci. It's not common to see foci-specific clones, except for one case example here. Okay. On the right side image, what we're doing is in trying to assess the degree to which there are differences or similarity in the genome, in the genetic architecture, in the clonal architecture of every single one of our cancer populations. And so here we're reporting the Jensen-Shannon divergence index on a scale of 0 to 1, 0 implying that the clonal architecture is the same between two malignant populations, a value of 1 indicating that the clonal architectures are completely different between one population and another. And as you can see for this patient, the values are extremely low. In other words, it doesn't really matter what your genotype is. Pretty much all genotypes are in position to create one of these populations. There's a high degree of flexibility in each of these genotypes to give rise to multiple phenotypes. Another way to look at this data is depicted here, where we're reporting the proportion of cells, the proportion of genotypes, across any individual malignant population. And so, for instance, a population here on the x-axis and the clones are on the y. So for each population, we're reporting the proportion of cells that come from one clone as opposed to another clone. And as you can see in the case of these populations, pretty much all of them have been arising from just about every single one of the clones we could identify in the tumor. Now, this is from one patient. If we expand to all other patients, of course, then we start to see divergence in this pattern. So there's another tumor that behaves exactly the same. But then there's a few case examples where, yes, we did find a genotype restricted to one foci that define, in this case, 3 different populations. And so this can happen, and this in combination with the heterogeneity we report across foci argues in favor when moving forward in studies that deal with patient samples to try to collect as many foci as possible and then perform your single cell technology to capture heterogeneity, as opposed to collecting one biopsy and going deeper in the sequencing of that one biopsy. All right, so intra-tumor heterogeneity is a little bit less than inter-foci, so we need to collect as many foci as possible when running these sorts of studies. What about when the tumor exits the primary site? So for our patients, for 4 of our 5 patients, we had also the opportunity to collect local regional lymph nodes from these prostate cancer patients, and could therefore start comparing the identity of the cells in the lymph node to the identity of the cells in the primary tumor. And so what I'm depicting you here is the UMAP looking at tumors in pale blue versus lymph node-derived cells in orange. And on the right side, looking at all of the cells coming out from these samples and looking at their genetic composition, assessing again here with the Jensen-Shannon divergence index, the degree of similarity or dissimilarity between cells coming from these different tissues at the clonal level. And this patient had a foci-specific clone in tumor 5, so that one is completely absent from the lymph node. So all the yellow ones have a divergence index that's very high, but cells or clones coming from all other tumor sites actually made their way into the lymph nodes, hence why the rest of the area has a very pale color score for the clonal architecture differences. And so this is more or less depicting a fact that what ends up in the local regional lymph node is pretty much everything that you can find in the tumor. Those lymph nodes are not imposing a clonal bottleneck. Instead, they're more keen to reflecting a discharge from the primary site, making it into these local regional lymph nodes. When comparing the population similarity of the populations we find in the lymph nodes versus the populations we find in the tumor, here specifically, again, looking at the cancer cells, here is what we observe. So for each of the 4 patients, the blue dots here more or less depicts what I showed you before, which is the degree of similarity between malignant cells across foci within a primary tumor. In orange on the left side, we're looking at the degree of correlation, of Pearson correlation in identity of the populations we can find in the lymph node. And so overall, we're in the same ballpark. 0.6 to 0.8 is the average degree of Pearson correlation we see within lymph nodes or within tumor sites. Now, when we compare lymph nodes versus tumor, this is the pink area of comparison. What we see is that the correlation drops compared to what we have in the lymph nodes in pretty much all instances. And so this is telling us that what this is telling us is that the cells that ultimately make it into the lymph node, while still having a correlation that's around 0.7, which is still higher than what one sees when we compare cells of different identity, still there are changes occurring in the lymph nodes. And so we wanted to identify and characterize these changes. And so what we did was to look at the nature of the cancer cells in the lymph nodes versus the nature of the cancer cells in the primary site, and asked whether there were any specific differences. We did so by looking at the RNA separately from the ATAC. And so here on the left side, we're specifically reporting the changes reported from an RNA-based analysis, so looking at differential expression. And what was identified was that the cancer cells that make it into the lymph nodes, once they reach that site, adopt an inflammatory-like identity. Many of the pathways associated with inflammation turn on in these cancer cells once they make it into the lymph node. And at the bottom, these are prostate-specific types of signatures that go down slightly that are very much more enriched within the primary site. From a chromatin-based approach, looking at these changes, similar things were observed. Down at the bottom are the DNA sequences that we tend to see enriched within the accessible chromatin of the primary tumor site cells. And what we find are motifs, for instance, for the forkhead factor and nuclear receptor, which is pretty much the androgen receptor, as well as the FOXA1, FOXA2 forkhead factors, which are known lineage-specific transcription factors of the prostate. While in the lymph nodes, we have a switch where the state is allowing for instead the activation of ETS factor and ETS factor, such that those are more associated with an inflammation signature. And just to put context of relevance in a clinical setting, to no surprise, this inflammatory-like state that we detect in the local regional lymph nodes and the cancer cells that make it into the local regional lymph nodes turn on in patients when you compare what it's like in the pretreatment condition at the gene expression level versus upon first-line therapy versus combination therapy versus ultimately the recurrence in patients. And so this signal that we see just as the tumor cells are exiting the primary site and entering the lymph node, the local regional lymph node, is already a signature that is associated with progression in these patients. And so from this first part of the talk, what I've been able to explore is the degree to which one can look at chromatin variants and epigenetic features to better understand the principles of epigenetic heterogeneity within a primary tumor context, as well as when the cells are exiting that primary site and entering the most proximal area, the local regional lymph nodes, we see a transition in cell identity. It is not a black and white transition, but it is one that is on par with a further progression towards a more aggressive phenotype. Okay, good. So that is for what goes on in the primary tumor context. So what about under metastasis? So prostate cancer, when treated, will often still develop resistance, and these metastatic resistant tumors will therefore be resistant to standard of care therapy, which is a chemical castration. And so we typically refer to metastatic prostate cancer as metastatic castration-resistant prostate cancer. To be able to study metastatic castration-resistant prostate cancer, or mCRPC in short, we had the opportunity to change cohort and work with our colleagues in California, specifically working on the Stand Up to Cancer West Coast Dream Team mCRPC cohort. That cohort is very appealing because it's been around for a while, such that whole genome sequencing has already been generated on the samples that we could access. Gene expression from an RNA-seq was also available. 3D genome organization measured through a Hi-C method had been generated, and ATAC-seq data was also available on pretty much the same set of samples. And so with those data sets, we decided to move forward in trying to identify what is the heterogeneity at the epigenetic level in these tumors, and how can it help us better understand the mechanisms that lead to progression. Now, one aspect that is often not mentioned when we look at any sequencing type of data is that we always have the opportunity to look at data from multiple angles. The most specific angle of relevant to this talk is that we can focus on the sequences that come from the non-repetitive genome, or if we are working with the right genome build, can expand our analysis to start looking at the repetitive sequences, which are primarily composed of transposable elements, as well as single repeats and the likes. And so for the next few slides, I'll show you how we've been able to study metastatic castration-resistant prostate cancer focused on the non-repeat sequences, and then subsequently on how we've been able to do so focusing on the repeat sequences. So for the non-repeat sequence analysis, Hunak was responsible for the work. And again, this was done in partnership with our collaborators in California. This work, which was published a little while ago, actually identified, based on classification using chromatin accessibility, 3 different clusters of metastatic castration-resistant prostate cancer. And in further characterizing these 3 clusters, what was identified was that 2 of them were reflective of elevated dependency on the androgen receptor, which is a potent oncogene in prostate cancer. And a 3rd group was actually weakly dependent on the androgen receptor biology. Instead, that 3rd group was heavily typical of what we refer to as neuroendocrine prostate cancer, so a prostate cancer progression phase that actually occurs by dismissing the necessity for androgen receptor signaling. And this classification from the chromatin accessibility data in these 3 different groups was more or less aligned with the classification one observes from gene expression. So if we go back to this left side image, this row here that's listed as mCRPC subtype actually is the subtype based on gene expression. And what you can see is that there's a series of samples here labeled as NEPC from gene expression that indeed fall within the NEPC cluster. We do not have NEPC positive samples except for one mapping to the other clusters. These AR high dark red tend to fall within the AR high attack-seek signature, and similarly the AR or the AR low map to the AR low attack signature. So a very high degree of alignment and agreement between the gene expression and the chromatin accessibility signature when assessed over the non-repetitive genome. But what about the repetitive genome? So this is work that was led by Lisanne Mutt in my group. And it was of interest to us based on a recent publication that had looked at chromatin variants over repetitive sequences in another model, in normal hematopoiesis, as well as in leukemia, where cells that have high potency, whether it's the stem cells or progenitor cells versus mature cells, have previously been well characterized. And in that prior study that came out earlier this year, what was observed was that across these stem cells and pluripotent cells in red and in orange versus mature populations, the overlap or the signal of accessibility over repetitive sequences was pretty much black and white distinguishing cells with high potency, with high capacity to give rise to diverse populations versus mature sulfate-committed populations. So these stem and progenitor populations were always associated with a high degree of accessibility over repetitive sequences here depicted in red. And this is across 4 regions in the manuscript. If you take a look at it, you'll see that this was done genome-wide. And it's a pretty impressive black and white type of stratification that one observes. And so we wondered whether if applying the same concept in prostate cancer would enable us to identify cases, patients, tumors, that were more likely to be associated with a high degree of plasticity, that were more likely to be associated with some form of tumor-initiating population. And so here what I'm depicting is the signal of chromatin accessibility across all repetitive sequences, all transposable elements that could be assessed, identifying a set of 605 transposable elements whose enrichment over chromatin accessibility differed sufficiently to classify our metastatic castration-resistant prostate tumors into 4 subgroups that were labeled here, line 1 for the red group, constant for the dark blue group, settle for the pale blue group on the far left, and then sign 1 plus for the green group. These labels are coming from us, and we gave those labels plain and simply because that first group that was labeled as line 1 plus has lots of transposable elements uniquely accessible in that group of patients, and most of them fall within the line category of transposable elements. The constant was called constants because very few transposable elements were found in accessible chromatin in these patients. The settle were labeled as settle because they contained a mixture of sign, line, ERVs, and transposons. So we formulated the terminology settle. And then the last one was labeled as sign 1 plus because, again, here, of all the TEs preferentially accessible in that subgroup of patients, the majority of them were sign 1 plus. Now, of interest in this bottom right figure, what I'm showing you is the relationship or the agreement or disagreement, actually, I should say, between this 4 subgroup classification that comes from accessibility over repetitive sequences versus the color labeled classification that comes from the gene expression. So suddenly we see, for instance, the green NEPCs being distributed across 2 groups, the blue distributed multiple groups, and a mixture of high AR, low AR activity transcription signatures across all of our 4 subgroups. So far from a one-to-one agreeing upon classification, arguing in favor of the signal over repetitive DNA, giving you a different biology, a complementary biology to what can be picked up from the non-repetitive genome. Of importance, our classification is directly related to outcome. And so here I'm showing you the outcome, the Kaplan-Meier curves for the line 1 plus samples, the constant samples, the settle, actually patients, I should say, and the sign 1 plus patients, based on their response pattern to ARSI, androgen receptor selective inhibitors, which is pretty much the standard of care practice to treat these metastatic castration-resistant prostate tumor samples. And as you can see, the line 1s are intrinsically resistant to this therapy, while all other subgroups show a degree of response to that therapy, arguing that the line 1s are special, that the line 1s require further investigation in order to identify alternative route to therapy. And so we took advantage of all the other data types that we had from these samples, so specifically whole genome sequencing, looking at copy number gains, copy number loss for a number of different genes. And what we discovered was a very strong and statistically significant association for the AR, the androgen receptor locus amplification at high levels within the line 1+ samples. This is further depicted on the right side, looking at copy numbers versus the line 1+ on the far left, and the 3 other subgroups that follow. So as you can see, we even have cases of over 150 copies, over 50 copies, over 25 copies of the AR gene locus. And so for those that are familiar with these types of high levels of amplification, you're typically already wondering, well, could there be some form of extra chromosomal DNA amplification for the androgen receptor locus because of these that could justify these high degrees of amplification? And so from the whole genome sequencing data, we could analyze it with the tool Amplicon Architect to identify presence of ecDNA status. Again, here stratified for our 4 subgroups defined from the accessibility over repeat sequences. And what we saw was indeed a biased presence of ecDNA positive tumors within the line 1+ cohort in dark purple. In pale purple, break fusion bridges, which are typical of what precedes the formation of ecDNA. And then less than 40% of cases with linear amplification or undetectable types of amplification. Now, the key take home from this here is that while there's often a huge push to looking at genetics to classify samples and to help us guide treatment strategy, if one had specifically restricted the analysis to genetics and specifically to the ecDNA positive negative identity, well, it's obvious from this figure that you would have missed the proper classification, because there's a series of ecDNA positive samples that are line 1+, absolutely. But you can also find ecDNA positive samples in the other subgroups, which argues that these have a different chromatin context despite having the same genetic event. In terms of expression, being part of the line 1+ implies that you are overexpressing the androgen receptor gene compared to other patient samples that are of different classification. When looking genome-wide at where these regions of accessibility over repetitive sequences were found, looking across all chromosomes, it was very appealing to us to see that within the line 1+ group, there was a skewed or preferential presence of accessible repetitive sequences on chromosome X. Those of you familiar with prostate biology will be excited by this discovery for the simple reason that this is exactly where the androgen receptor is found. This is the androgen receptor location on chromosome X. On the left side, I'm showing you chromosome 8, where MYC oncogene is located. And then for each of the patients from the line 1+ cohort, I'm depicting the density of accessible repetitive sequences. And while it's pretty much uniform across chromosome X around the MYC locus and beyond, what one can see on chromosome X is that there is a high degree of clustering of accessible repetitive sequences right on top of the androgen receptor locus. The fact that we had identified our regions using chromatin accessibility, looking at ATAC data, implies that we have a tendency to find these DNA sequences that are preferentially hotspots of recruitment for transcription factors. And so we therefore decided to assess whether these repetitive sequences being accessible in the line 1+ cohort would serve as docking sites for transcription factors. And so for this, we took advantage of the REMAP database that contains thousands of ChIP-seq tracks for different proteins across different cell and tissue types to assess enrichment of these ChIP-seq tracks over the accessible repetitive sequences in our tumors. And here are our top lists. So for those of you, again, familiar with prostate biology, you'll be excited to see FOXA1, AR, TLE3, OXB13 as our top hits. These are extremely well-known lineage-specific transcription factors. Zooming in at the higher resolution where we're looking at individual transposable element families here, looking at the enrichment for AR, FOXA1, OXB13, and TLE3, you can actually identify which repetitive families have the strongest association to recruit these transcription factors in prostate tumors. Now, another aspect that we could exploit was the fact that we had high-C data so that we could assess whether these preferentially accessible repetitive sequences in the line 1+ patients were found at the AR locus, not just as passenger events, but in order to control expression of the androgen receptor. And so what we could do is specifically use the high-C data to assess the degree to which these repetitive sequences were looping to the promoter of the androgen receptor. And so on the left side, I'm showing you the results from line 1 patients. And on the right side, I'm showing you the results from another type of metastatic castration-resistant prostate tumor, the sign 1+. All the black lines come out from the AR promoter and go to a distal site that is not repetitive, but accessible. And all the red lines go out from the AR promoter to a distal site that is accessible and harbors a repetitive sequence. And so to no surprise, there's no red lines in the sign 1+, but there are hundreds of red lines in the line 1+, 548 to be specific, which demonstrates that these accessible repetitive sequences, at least 500 plus of them, are actually physically interacting with the androgen receptor promoter. And so this leads us to this following model. In these patients that are intrinsically resistant to castration-resistant therapy, so to this androgen receptor selective inhibitors, these line 1+ are in a position to overexpress the androgen receptor because they have acquired chromatin variants over repetitive sequences that turn these repetitive sequences on as regulatory elements that can interact with the promoter of the androgen receptor to enhance its expression, to induce its overexpression. And of importance, these distal sites are actually binding sites for the same product of the gene being overexpressed, i.e. binding sites for the androgen receptor. So therefore creating a positive feedback loop within these regions of amplification. Other MCRPCs have baseline levels of androgen receptor and therefore are in a position to respond to the drug because they do not oversaturate the delivery of the drug. These can compete out the drug because there's enough AR molecule that some will be free of the drug in order to be able to promote signaling and growth of the cancer cells. And so the implications are the following. This concept of heterogeneity, in this case, reported at the interpatient level, allows you to, at the endpoint, to impose yourself as a metastatic population, to adopt one of multiple different fates, some of which by default making you intrinsically resistant to standard of care therapy with these androgen receptor selective inhibitors. All right, so to conclude, once again, I'll remind you of the folks that did the work from my group. Lisanne did the work that was presented with regards to the metastatic castration-resistant tumors. Kira and Tina were involved in the primary tumor assessment. In collaboration with colleagues at the Princess Margaret Cancer Center, Hanson He and others, and our colleagues in Europe, whether it be at the DKFZ in Germany, in Copenhagen, in Denmark, and Berlin, in Amsterdam, and Hamburg, as well as colleagues from the US and Minnesota, Wisconsin, Fred Hutch, the Dana-Farber, University of Washington, and the team out west, Felix Fang and David Quigley at UCSF. So with this, I'll thank you for your attention, and I hope we can pursue this conversation in better understanding the premise of epigenetic heterogeneity as a driver of plasticity and tumor progression in the minutes to come. Thank you.
Thank you again, Dr. Lupien, for the great walkthrough. And we'll now move into the Q&A portion of the session. And we've received a few questions throughout the webinar. Thank you for submitting them. Let's see. Michael, is there a question you want to ask first?
Yes, maybe ask my question directly. I'll put it in the chat. It's an awesome talk, Mathieu. I really enjoyed it. It's a bit selfish being on the panel asking the first question. The question I have is, right, we all saw that Halda Therapeutic got acquired for a huge amount of money, and mainly for their AR-BOD4 induced proximity molecule. I'm wondering, given your expertise, if you can speculate on possible MOAs, because in the public domain it was described as a halt and kill mechanism. But now with your data, I'm wondering what you think the mode of action is for that molecule.
So that's an interesting question because it goes outside from what we've been able to test so far. So I'm not 100% committed to providing an answer before we play with it ourselves. But the concept of coming up, but what I can say is that the concept of wanting to target the androgen receptor and come up with alternative methodologies is definitely worth considering, whether it's through a degradation pathway system, whether it's by targeting the promoter of the androgen receptor. I think those are all valid options. We'll see which ones work for which subtypes. The key thing is I think we need to be fully aware of which subtype we're working with in order to move forward. So for instance, in our case examples of the line 1 plus, a degrader or an antagonist, as has been used before, is probably going to be there's probably going to be intrinsic resistance. However, if one was in a position to specifically target the promoter of the androgen receptor, which is the point of convergence from all these signals, I'm hopeful, but again, I have no data, so I cannot confirm, but I'm hopeful that that will turn out to be more effective.
Okay. Thank you for the answer. I have a couple more questions. I go off camera.
Thank you, Michael. Thank you, Dr. Lupien. Our next question, Dr. Lupien, you showed there's a link between specific types of cancer, for example, colon cancer, and chromatin-specific variants. Do you see any similar link between chromatin variants and cancer-linked mutations, such as KRAS mutation?
Yeah, absolutely. So I would actually push that further. So I'll explain that in two aspects. So what we've shown and others is that the actual accumulation of mutations in the tumors typically is skewed towards areas that matter to that cell. And so since the epigenome is different from in the normal tissue versus the cancer tissue, the tumor, often the mutations make sense when you study them in the context of the chromatin in the tumor, but are harder to explain when looked at in the context of chromatin in the normal tissue. So we had a study in partnership with Wilbert-Svart and prostate cancer on that topic a few years back that I would recommend as a primary source of exploration. The second aspect to that question that I would put forward is the following. If a mutation is currently active, if a mutation is currently, to use the terminology, driving the cancer phenotype, my expectation is that it will alter the chromatin, okay? Such that we could actually have that as a criteria beyond just using the mutation frequency and the density of mutations to be able to identify, let's say, cancer drivers. I would be a strong advocate that in addition to be able to say, is it a driver now or was it a driver in the past, that we be able to demonstrate that these mutations are changing the chromatin because they need to have a long-lasting, inheritable change in order for their phenotype to be sustained. And a mutation does not work alone. So if a mutation is to be a driver mutation, it should also impact hundreds, if not thousands, of genomic coordinates and changing their chromatin state, because that's what's required for transformation. You won't be able to transform a cell into a cancer cell by changing the chromatin state at a single DNA sequence. It's the same principle as what was depicted for mutations in the past. You need a series of mutations to have a cumulative effect. If you're talking specifically of a given KRAS mutation, well, if you want that mutation to have a long-lasting effect, it has to lead to changes in the chromatin that are also themselves part of the KRAS mutation package.
Thank you. Our second question, the mechanism of alterations in chromatin accessibility in different population of tumors could be due to changes in the activity of various chromatin remodelers. Have you compared the heterogeneous tumors with different activity of chromatin remodelers?
Yeah, absolutely. Good question. And we could actually push it a layer above, which would be to look at the microenvironment. And one can actually be very specific to look at regional changes, differences in the abundance of specific metabolites. So for epigenetic modifications to be added onto the chromatin, whether we talk about methylation, for instance, we need the metabolite SAM, S-adenosyl methionine. It is the universal methyl group donor, and the abundance and levels of SAM are definitely fine-tuned, regulated, and readily affected by diet and the likes. For acetylation, acetyl-CoA is needed. For phosphorylation, right? ATP is needed, and so on and so forth. So one could consider that indeed there are these regional populations of cancer cells that one can pick up because there are regional variability in the abundance of such metabolites, which would impact the activity of the chromatin modifiers, right? So that's a short answer. The long answer is if you think of all the mechanisms through which chromatin states can occur, can change, well, definitely metabolism is one, environmental insults, mutations is another. And then the third one that we haven't discussed so far is stochastic changes, error introduced during the process of replication that would mimic the error rate, for instance, that one sees in the DNA sequence, again, in the process of replication.
And from the same participant, we have a follow-up. We're asking, were you able to capture unique accessible signatures across hybrid tumors?
Across hybrid tumors, what is the definition of a hybrid tumor? Might be what I need, what I can say. So it might take a while for the participants. So happy to connect after the meeting if you send me an email. But what I can say, hopefully that's going to help addressing that question, but I'm not sure it will be, is moving forward, we're transitioning into the spatial technologies in a way that will allow us not only to identify the different populations that may lie in one site as opposed to another, but at the same time, pair that up with additional layers, specifically layers that will look at metabolic differences to see if we can actually draw the links between population A, metabolism A, versus population B, metabolism B.
Thank you, Dr. Lupien. And our next question, they said, fascinating work. Did you find a psychological setting where the many L1s at the AR locus are active as in cancer?
So if we found, just rephrase that question, sorry, if we found
a physiological setting where the
line ones, yeah.
At the...
Not yet. So if we look back at the work that was done in hematopoiesis, where in hematopoiesis they have access to high resolution purified normal populations, such that they can truly study hematopoietic stem cells, progenitors and the likes. We don't have that in prostate yet. So the normal prostate epigenome has not yet been released, at least not to the point where we're comfortable with it, and surely not to the point where we can identify prostate stem cells, for instance, if we want to use that word. I hope I'm not like disturbing too many people by making that terminology, or any other type of prostate progenitor populations. But the idea is to be able to look at these higher resolution normal populations to see if the line ones are turned on. So is it that the prostate tumor hijacks a mechanism from an earlier, more primitive prostate population? Or is it really something that's cancer specific? That we need to assess. But if you go into the recent hematopoiesis publication that I mentioned on one slide, there it does do the comparison with leukemia. And the differences between the leukemia stem cells and the normal hematopoietic stem cell is very minute. There's a series of only 21 transposable elements that are unique to the cancer setting. So it's a very small subgroup.
Awesome. And our next question, great presentation. I had a question regarding MCRP work. You found AR formed a feed-forward loop, which is reminiscent of core regulatory circuitries. Did you see other transcription factors that also bound to the same AR areas that were also activated by repeat elements? And a quick follow-up, would those core regulatory partners be a better target?
Okay, good. So the first part, the answer is yes. So FOXA1, OXP13 are typical factors that go to the same coordinates as AR and work together, precede to make the area even more permissive for AR binding and the likes. So the answer is yes, and we tend to be looking at factors that are highly related to one another, not necessarily in a complex, but in a sequential order of recruitment. Now, in terms of treatment strategy, the key issue is that all the factors we've identified so far are nuclear factors, so factors that preferentially lie within the nucleus. And by default, targeting these factors is extraordinarily challenging. It's always been challenging. We're fortunate with the androgen receptor itself being a steroid receptor that we can use anti-steroids, which will cross the membranes, whether it's the cytoplasmic or nuclear membranes, without any issues. But for any other type of drug, it's a challenge. So should we keep moving forward and developing chemical-based compounds to antagonize these factors, or should we think differently? I'd like to think differently. And so there's beautiful work and more to come that has tapped into the biology and acceptation of RNA medicine that followed up on the COVID vaccines, the mRNA COVID vaccines, such that now there's strategies to do delivery of RNA and potentially even of single-stranded DNA that could be targeting sequences in the genome of the cells of interest as opposed to targeting proteins. And while it's too early to say it's going to work, it's too early to be committed to success. There's definitely a high interest in my group to be able to explore those types of technologies for the next generation of therapy that could actually target transcription factors, not in a protein context, but rather in their expression, either targeting their promoters, targeting their distal regulatory elements, so therefore targeting the chromatin at specific DNA sequences. Because that works in the lab, right? When you use CRISPR technologies to change the chromatin state, so CRISPR-I, CRISPR-A, and derivatives, that works in a model system. Now the key is to be able to transfer that capacity into an in vivo context.
Awesome. And our next question, is this epigenetic heterogeneity specific to solid state tumors? Have you done any assays in liquid state tumors like AML, or if you can speculate what mechanisms would be more common between those two?
Yeah, no, this is definitely not specific to solid tumors. It applies to liquid biopsies. I would argue actually that it's most interesting to look at these mechanisms versus the genetic, like versus the mutations in childhood cancer versus adult cancer, based on the premise that adults are exposed to much more mutagens and age-associated mutations accumulating over time, while the pediatric cases are typically free of mutations. So I think that if you want to look at demonstrating a greater role of epigenetic changes in the process of oncogenesis, it's not so much solid versus liquid tumors, it's rather going to be pediatric versus adult.
Awesome. Our next question, thank you again for the great presentation. From a researcher at Crimson Global Academy, I was wondering if there were any standout individual or a combination of epigenetic marks that were associated with the accessibility changes in the various patients' metastatic tumors at the promoter or other loci.
Perfect. That's an interesting question because it draws from the fact that there is a degree of diversity that is possible within the chromatin states that far exceeds what is possible from the DNA sequence perspective. And there was that one slide that I showed all these different chromatin states from different combinations of histone modifications. The problem is when you move into patient samples, you're restricted into the amount of material you have access to. So typically you'll get like a small chunk, or even worse, you'll get like a 10 micron thick slice of 2 millimeter in diameter. And so the availability of material for multi-assays is close to zero. So you have to choose one assay as opposed to another. So we chose chromatin accessibility for a series of different reasons. The premise is one, it's easy, it's fast, it works on a very small number of cells, like 500 cells is enough, like a 10 micron thick section is enough. And from an epigenetic standpoint, from an annotation standpoint, it allows you to measure what's going on at multiple different types of DNA elements. So you do find promoters, you do find enhancers, you do find insulators. The shape of the accessibility will distinguish a promoter from an enhancer, will also distinguish an enhancer from an insulator, and so on and so forth. So there's a lot of information embedded within chromatin accessibility data. And so we weren't able to do, let's say, 5, 10 different histone modifications to gain a degree of resolution that would be promoter specific, enhancer specific, or anything else. But we're using ATAC as a surrogate. And because we're working with patients, that's what we can do. If you work with model systems, you can go crazy and do everything. And there I recommend people to rely on the recommendations from the IHEC, the International Human Epigenomics Consortium, which has 6 specific histone modifications that it proposes that capture as much as possible the comprehensive types of DNA element that you can report from histone modification. But if you do that, you'll miss out on the more recent modifications that colleagues are discovering that would be specific to unique environmental conditions that have to do with high glycolysis, for instance, or hypoxia, and so on and so forth. So it's not an easy decision to make, but happy to take that conversation offline by email. Feel free to follow up by email if you want more discussion on the question.
Lovely. We have two more questions here.
Okay.
So the first one, do you see the chromatin states differences among different metastatic cells of different cancers? Is it possible the tumor niche is promoting the chromatin state changes, and can this be predictable among different cancers?
So, three multiple questions. So, chromatin accessibility differences, yes. Between patients as much as between cancer types, yes. Can this be predictable? I'll focus on that aspect of the question. Yes, but we need data. At this stage, we lack a comprehensive international effort to systematically, at high resolution, profile from an epigenetic layer, as well as from a gene expression layer, as well as from a mutation layer, as well as from a metabolomic layer, the complexity of a given tumor to be able to come up with proper predictions, in my opinion. Like the ideal is to more or less revisit the TCGA if we want to, and add these extra layers at single cell resolution for metabolism, epigenome, genome, and expression. Those are the minimum that I think are critical to be able to move forward with good prediction of heterogeneity based on not necessarily always being able to collect all of these data sets.
Thank you. And our last question here, they're curious if you or your colleagues have seen intra or even intertumoral immune-related dysfunction, modulation, changes driven by or associated with chromatin differences, etc.
Perfect. So in the samples we collected, there was actually a selection for samples that had a higher cellularity content for cancer cells. And so, for instance, in the primary prostate tumor context, I did point out that we could identify some normal populations, but the number of cells in the normal was still small. So we need to, moving forward, be more permissive of having a 50-50 proportion of cancer cells versus normal cells in order to really in-depth address this question. What I can say for now is something very interesting, is that the normal populations amongst those that we could collect, and that's part of the primary prostate study that I showed, the only population that showed a high degree of patient specificity in the normal cells were the B cells. And so that's very interesting when you consider exactly that these patients are all lymph node positive. And so the premise is that we have our cancer cells exiting the primary site and entering the lymph nodes. And in that context, the expectation is that these cancer cells would be in a position to effectively interact with the B cell so as to influence the B cells in the lymph node and somehow impact the B cell composition in the primary setting. Now, this is just from a handful of samples, right? So we need to be more comprehensive in this analysis. But we did notice that the B cells showcase the highest degree. It's the only population of the normal populations we could find that showcase a degree of patient specificity. And again, these patients are lymph node positive. So if we had lymph node negative patients, would we still see the B cells classify as patient specific? If the answer is no, then I think it implies that this initial migration to the lymph node is indicative of sending a signal that allows the B cells to be paired with the cancer cells for having a patient-specific identity. If our lymph node negative B cells are also patient-specific, well, then that's the false hypothesis, so we'll have to think of something else. But at this stage, it's too early. It's just hypothesis. But yeah, B cells is what if we were to go in that direction of looking at the immune composition being patient-specific and interacting with the cancer cells, the B cells would be the first population of cells we would pay attention to.
Thank you, Dr. Lupien, for staying over a bit and answer these amazing questions. So that brings us to the end of today's webinar. I do want to thank Dr. Lupien again for sharing your insights, and thank all of you for joining us today. And we hope to see you again at a future webinar. And have a great rest of your day. Thank you, Dr. Lupien.
Thank you. Have a great one.